Thanks very much, Ella, and hi everybody. I'll just see if I can get this to work. Big green arrows go forward, is it? All right, so yeah, my talk is all about blueberry rust, obviously, and potential survival over winter. And when I say winter, obviously winter is different for everyone depending on where you are. This project's funded by the Tasmanian government, so we're very much focused on Tasmanian winter. However, we are also collaborating with people outside of Tasmania. So we've got a team at Southern Cross Uni, Jay Anderson and Karina Griffin as well there too, and also this is part of a larger project where we've got a whole team of people involved. So yeah, we've had some great contributions. So obviously, blueberry rust is caused by a fungus. It's got a fairly new revised name, which is Puciniastrum minimum. Those of you who've seen it in the field will know that it's got a kind of a red lesion, and on the top side of the leaf it's quite red. On the underside of the leaf, if it's an actively sporulating period, you'll see these yellow spores, and they are highly transferable. They can be distributed very easily. You can also get chlorosis and premature leaf drop once that disease becomes quite extensive. It's been present in Australia for over 20 years, and I think every state in Australia has it now apart from South Australia. And in Tasmania it's been present now for some years since the original incursion, and then a second incursion, a very big concerted effort to try to contain it to infected premises for a number of years by Biosecurity Tasmania, and then eventually it was deemed endemic in mid-2022. In fact, I think that happened the week after the last berry quest. I remember being up there in the Gold Coast and hearing that it was just about to be declared endemic. So it seems like a long time ago. So with disease management, you know, with a lot of sort of, I guess, airborne and foliar diseases, there's some key principles that are important, and rust is no exception. So things like reducing canopy moisture, making sure there's good air movement through a crop so that you don't have wet leaves that will then, you know, stimulate those spores to germinate and infect. And so pruning's useful for that, obviously avoiding overhead irrigation and row orientation. Monitoring for presence of the disease is important, and then of course there's a whole range of fungicides that are available. So on the right there you can see the list of those currently registered for control of blueberry rust in Australia, both conventional and organically approved. And this is our second project funded by the Tasmanian State Government on blueberry rust. In our first project we were looking at comparing a range of organic or potential fungicides for control of blueberry rust, and that was actually done in partnership with New South Wales DPI. And I've got Melinda here, who was a big part of that project, and we were able to show that Intervene had some ability to suppress the rust. So that was then added. We got a minor use permit for that, and it was then also added to the ICA 31 to enable organic growers to have two products that they could then use to meet those interstate certification to you know, move fruit into areas that didn't have blueberry rust and access markets, for example, South Australian market. So yeah, there are a range of fungicides, of course, but with any disease management, using them you know, as little as possible is ideal, and so therefore understanding more about the risk, the prevalence is important. So inoculum removal is really important. The more that we can reduce the build-up of inoculum, the less disease there will be, and therefore also the less fungicides that will need to be used. So obviously blueberries come in all shapes and sizes. We've got those that are deciduous and will defoliate over winter, and in semi-deciduous and evergreens obviously retain leaves over winter. So I guess the question is, for us, is that, well, first of all, we could say that evergreens will obviously retain the rust quite happily over winter in warmer environments, and it will stay active, you know, probably most of the year. But what about colder temperatures? So what about in, say, some of the colder parts of Tasmania? Do we think that varieties that are holding their leaves over winter? Do we think that the rust will survive through the winter, or might it not? And if it doesn't survive, is something like artificial defoliation a useful tool that could be added to the list, either to, instead of or as well as using fungicide treatments too? And I guess these two questions are the basis of the talk that I'll be going through today. And these two questions actually came out, I mean, they're good general questions to understand in terms of the rust life cycle, but they particularly were born out of decisions that Biosecurity Tasmania were making at the time of the containment strategy. So trying to decide, if we had infected premises, could we have some understanding of the options for those infected premises to be resolved of that status, I suppose. So ensuring that, the rust had not been observed on that property for 12, 18 months' time, and could we add in some, buffers to say, well, it's a very cold site, therefore we expect it wouldn't be surviving. So some knowledge around those kind of factors was of interest at the time. Obviously, we don't have that containment plan in place anymore in Tasmania, so the relevance of the questions is slightly pushed to the side, but we still think it's really of interest to understand what we're dealing with the rust pathogen. So rust pathogens have very complex life cycles, and blueberry rust in its home environment, which is sort of in Northern America and Japan, has all 5 rust spore types, and this is because it has 2 host plants available that allow it to complete its life cycle. In Australia, it's a much more simple situation because the only spore stage detected is the Eurydinia spores, and you can see them up there. Now, it's interesting when you see that list of different spore types, you see the top one, Telia spores. So in its home environment in the northern USA, these Telia spores allow the pathogen to survive over winter. They become dormant and they allow the pathogen cycle to kick-start again in spring. But in Australia, we don't have that. So we might assume, well, it can't survive winter very well. It doesn't have those spore types, but it may be that the Eurydinia spores here are sort of adapted, a bit more cold adapted, because it's been here and sort of having a different type of life cycle. So the other point to make for those who aren't familiar with the rust is that it will only survive on living plant tissue. So if leaves are defoliated and dropped to the orchard floor, within 2 or 3 weeks they will basically stop being a source of inoculum, and particularly in warmer environments where those leaves will break down really quickly. So the issue really is around living leaves and whether the pathogen survives on those. So what we've been, the questions we've had, and this work has been done in association with Southern Cross University. As I said, they've been delivering a subcontract on our project to work on these questions. So the questions really are, so we know that the pathogen survives on living leaves, but under what conditions, what temperature range could we expect to put some limits around it? And is there a minimum cold tolerance for the survival of those spores, the Eurydinia spores? There's been quite a bit in the literature about the temperature ranges that the spore type require to germinate, and particularly the optimal infection conditions, and that's really helpful when we're thinking about infection risk and so on, and timing of fungicides, for example. But we don't actually know what's actually going to kill them, what temperatures are cold enough to kill it. So the other question is, the spores have got to be produced from a pustule, so the structure on the leaf, that's the little infected mass on the leaf that starts to produce all the spores. So what can actually kill that little pustule on the leaf? And is there a difference between the spore survival and the pustule survival? So the team there at Southern Cross Uni have done a range of controlled trials during last year. And it took them quite a while to get the methods running for this, because it's really quite tricky to work with these systems and get good methods going. But what we did, we thought we'll go to -5. Surely it can't survive at -5. I mean, that's cold, right? So we thought we'd start with the coldest we thought it could survive. And then we went 0 degrees and +5. We exposed for 4, 8 or 16 hours and put the leaves in tubs like this and then put them in these controlled temperature chambers. So Karina and Jay then were able to assess spore viability and the germination. So with the viability, it's staining the spores with two different fluorescent stains, and then looking at them under the microscope. So many, many hours of microscope work there. And then also looking at germination of the spores onto a water agar. And again, many more hours of microscope work there. They also, as well as looking at that spore survival, they then looked at the pustule area and whether it remained active or not after those cold temperature exposures, and then whether the spores on those pustules remained viable and were able to germinate. And the experiment was conducted 3 times. So the results, I could say, we've got pages and pages of charts and figures and models on this, but I guess, shorten it right down, spore viability was not affected by temperature or exposure period. They were still viable, so they were still coming up as nice and green. So that figure there on the right is showing the fluorescence of spores under the microscope. So the green ones are the ones that are alive, stained with FDA, and the red ones are the dead ones. So between 90 to 100% viability still existing on those spores. And the germination was a little bit reduced by the freezing, but not affected at all by the other temperatures. So it's a hardy beast. These spores are very hardy. We then looked at the pustule development after the temperature treatments, and what this chart here shows you is we actually had 14 different treatments, and it's a bit complicated to explain, so I'll just zoom in on the key highlights of the data. But we had room temperature, and then the -5 for 4 hours, 0 for 4 hours, +5 for 4 hours, etc. And the P1 is the pustule counts per leaf immediately after the exposure of those temperatures, and P2 is 1 week after exposure. And I should note as well that you'll see in the photos the leaves are detached. So leaves are removed and then given a wet cotton wool kind of, wrapped around the petiole to try and keep those leaves alive, but acknowledging it's still an artificial situation. But 1 week after exposure, and then I'll just highlight those. So you see at -5, there's a much bigger drop in the number of pustules per leaf after the -5 treatment, whereas with the other temperatures it was a drop, but not nearly as substantial. And then the point at the bottom, 2 weeks after exposure, by that time point, there were no pustules observed on those leaves that had been treated at -5, but still some active ones on those treated at 0 and 5. So we're really seeing here that it takes a lot of cold to knock this pathogen out. So what we thought we'd do next is actually focus on the pustule development itself, because it really doesn't matter if those spores are still alive if the pustule is dead and is no longer producing those spores, if you know what I mean. So we thought we'd try again and focus more on pustule development, and this time look at whole plants, so have whole potted plants that are infected and then expose them to cold temperatures, have them acclimatised first so they're not getting completely shocked by the cold, and then look at if there's a difference with that survival on the cold temperatures, and using repeated cold temperature, sub-zero cold exposures to see what happens. But I think the punchline really is, not many areas of Tasmania even have -5 for repeated exposures. So we're assuming that the pathogen is just going to keep surviving in most cases. So also just matching this a little bit with observations, and we've only really just started doing this, and we'll do it more this coming year. Looking at just, for example, some potted plants that I had on the university campus just up here, Sandy Bay campus. We had some active rust pustules on legacy plants. The pustules were very much present. They became non-active over winter. So just looking at them through the winter, the lesions were still there, but there were no active pustules, no spores. So they sort of disappeared. And then come spring, they really seem to pop again. So that might be something that many of you are seeing in your areas if you are looking at rust, and you might be seeing it, kind of come and go. So be really interested to hear what people are seeing. If anyone has the time to track me down, that would be great. And also just comparing to a commercial orchard where we had active rust on a legacy cultivar in an unsprayed row. We monitored a particular number of plants and branches every week over winter and spring, and also looked at the temperature, and we found that that the disease was basically still there through that whole period as active rust pustules. So it got down to minus 0.1 degree, so they were still there. Perhaps if it had been a bit colder, they might have disappeared. So I guess that's where we're at with the temperature work. And what I'll move on to now is the work we've been doing with artificial defoliation. So this is, I suppose, an alternative to thinking, well, cold is going to mop this pathogen up. Well, if it's not, and if you don't want to use fungicides, or for some reason they're not effective or not accessible, could defoliation be an option if you really want to reduce that inoculum? And obviously defoliation can also be of interest for people wanting to change their fruit timing. So I guess it's of interest for that perspective as well. But we've done trials now over 3 years, and I'll show you the data for those today. So back in 2022, so all of these trials that I'll show you were all done on one property in southern Tasmania, and all with legacy, which is it down here is holding quite a large percentage of leaves over winter. So with legacy, we had a first trial which trialled the use of protone, which is a synthetic abscisic acid used mostly in grapevines for coloration. VBC, which is comprised of a compound called ACC, which is related to stimulating the ethylene biosynthetic pathway. And then we looked at zinc sulfate as well. So we've got two sort of plant growth regulator products, and one, nutrient that can certainly lead to those leaves being sort of becoming desiccated and falling off. So different modes of action. And in that trial, these were all applied in early July of that particular year. So I'll show you the results of that shortly. 2023, we moved on to looking at urea and a urea plus copper chelate option, and then a copper sulfate. And then last year we looked again, going back to protone, and we looked at 2 different timings of application as well. And in all of these trials, we have a control, obviously, and we compared to hand defoliation, because we wanted to see, well, what's the effect of just the removal of leaves in the absence of the actual other perhaps side effects that the chemistry might be causing. So to get straight to, I actually didn't show you the results of the 2022 trials. So with the 2022 trials, we actually found protone and VBC didn't have an effect at all. We didn't get hardly any defoliation, and it's probably because we applied it during midwinter. It was too cold, so protone really needs to be a bit warmer. And the zinc sulfate did do a great job of defoliation, but it reduced fruit yield by about half. So we didn't think that was worth pursuing. With the 2023 trials, we found that the defoliation was pretty good for the urea plus copper program, and the copper sulfate did a decent job. It knocked off most of the leaves, but not quite all. But then what was the effect on yield? So we found that hand defoliating really had no effect on yield, so you can remove the leaves, the plant can regrow those leaves and get enough carbohydrates to still produce a good crop. In fact, it's slightly higher there. But the urea and the copper sulphate had a massively reduced yield, so not really commercially inspiring. And we think that, so those programs had actually worked quite well in some deciduous varieties where people were just wanting to bring forward that defoliation. But in a semi-deciduous like Legacy, I think there's too much active growth still going on. There's flowering buds that are still active, and the chemicals have knocked them around too much. We then looked just a couple of months ago to see what the effect of the just the hand defoliation was a year later, and it was still no different. So you can remove leaves, but obviously picking them by hand is not economically efficient. Now that's just dropped off. All right, I'm almost done. The 2024 trials, we looked at protone again, hand defoliation. So we've got control, hand defoliation, protone. Removed leaves very, very well. However, we did get a drop again in yield, but much better than some of the other products. So yeah, we got significant effect of treatment on the yield, whether it be hand or protone, and that's a difference from the previous year. So we did get an effect of yield this time with the hand defoliation, but no significant difference between time 1 and 2. And just another point on timing, just looking at fruit production, yeah, the protone did result in a little bit of earlier fruit, but not substantially so. And I'll just jump straight to the summary points. So there's a few chemicals out there that can cause defoliation. It's difficult working with a variety like Legacy that's still actively growing. The point about the hand defoliation, so we think that the fact that we got impact on fruit from last year's trial but not the one before was that for last year's trial, we did the hand defoliation in autumn. So doing it earlier rather than doing it in midwinter might have made a difference there. And protone seems to be the most promising, but it is expensive and at the moment not commercially registered. So that's really the where we're at with those two major parts of what I wanted to talk about. There's a couple of other aspects of research going on in our project which I don't have time to talk about, so I'll just skip through those slides and really just thank everyone who's been involved in the project, thank the state government for the funding, Fruit Growers Tas, Biosecurity Tasmania, our orchard partners and all the staff that have been involved. Thank you. Oh, and a plug for Melinda's Managing Blueberry Rust and Guide, so get your hands on that.
Fantastic. Does anyone have any questions?
Thank you, that was really interesting. I was wondering, when you did the defoliation, you checked for yield. Could you also check for rust incidents on those plants in the next year, or is that, there's no rust in that orchard?
There's no rust in that orchard, yeah.
Okay.
Yep.
Thank you.
Any other questions?
Okay.
Yeah. Just wondering, have you considered mechanical defoliation at all, like a commercial leaf blower or something like that to remove leaves?
Hadn't considered it. Good question. I'm guessing it would have to be, do a very strong job to get those leaves off, unless you did a first treatment, say, of urea to just loosen them up and then blew the rest of them off. You might get less damage on the yield, less influence on the yield by doing a combination, perhaps. Great idea.
I'm just wondering, are there any other host plants for blueberry rust in Tasmania other than blueberry?
I don't believe any have been, the rust has been detected on any of them, but certainly rhododendrons are a host. There's a few other related species, but the main point is that we don't have the acial host, so we don't have the full life cycle.
Thanks.
Yeah.
Yep.